and welcome for the first time to Glycan Hub, the podcast in which we explore health, disease, and longevity through the lens of glycobiology. My name is Rina, and I'm your host. So let's get right into the first episode of this season. The immune system is the army of the body, armed with artillery which has allowed us to fend off foreign invaders for generations, and in the process evolved to distinguish between self and non-self, between the tissue of the body and the foreign intruders. However, there are instances, such as in cancer, where the immune system must be able to recognize that what was once a part of the system is suddenly posing a threat. Unfortunately, there are also times where the immune system might mistake healthy tissue as foreign and treat it accordingly. My guest today will introduce us to the intricacies of immune regulation and the role glycans play in the interaction and communication between the immune system and the cells of our body. We'll discuss how glycans on the surface of our cells act as ID cards, as well as how pathogens are able to mimic host glycans in order to evade detection and perpetuate infection. But the central question of the following conversation is what happens when the regulation of immune tolerance falters and the immune system fails to distinguish between friend and foe. My guest today is the Immunology, Cancer and GlycoMedicine Group leader at the Institute for Research and Innovation in Health at the University of Porto, as well as the affiliate professor at Faculty of Medicine. Her research focuses on investigating the role of post-translational modifications by glycosylation in the regulation of key proteins functions involved in cancer and chronic inflammatory conditions. A warm welcome to Salome Pinho.
Thank you. Thank you for the kind invitation.
Salome, I'm interested, first of all, how did you initially get involved into the field of glycobiology, and what is it about the field that has made you stick around and continue your research?
Actually, my very first contact with the glycobiology field was at the faculty, but at that time was not pretty much explored and about the relevance and the impact of the field, only just mentioning that proteins can be decorated with carbohydrates and we can have glycoproteins or we can have glycolipids. But in a more specific and in a deeper way, I was enrolled in the field in my PhD at the University of Porto. So one of my mentors at that time was Salomé, which kindly introduced me the glycobiology field. And from the very beginning, I got in love about the field and the many biological and clinical impact that glycans and protein glycosylation can have in health and life sciences in general.
Now, today our conversation is going to talk much about basically glycans as a language, essentially, in our body. And so I think a good place to start is by maybe having a bit of a visual representation. So most of the time when we look at illustrations of cells in our bodies, they are shown as very smooth on the outside, when in reality they are actually very fuzzy. So could you tell me what is this fuzz and what is its purpose?
The way that we have a visual insight on the surface of a cell, so normally we have them on textbooks, on electronic microscopy view, and actually we can appreciate that dense and complex coating of something, and something is glycans, are sugar chains that form that major biological and physical barrier that occurs between cells and environment. In fact, all of our cells from our body, but also from other organisms such as virus, bacteria, fungi, all of them are covered with this coating, this shield of glycan structures that arise in a cell surface, giving this visual impression of a kind of a forest that actually is this fuzzy aspect is because of the diversity of glycan structures that arise in a cell surface. Yeah, that's why they are so important.
Now, we are specifically going to be focusing on the role of the immune system and its ability to distinguish between what is home and what is foreign in our body. And this invites this idea of immunological tolerance. Could you explain what we mean by this and how our immune system is able to distinguish between what is self and what is not self?
So all of our cells, and actually most of the cells in nature, are covered with glycans. And glycans is actually an evolutionary process. And there is actually this huge diversity of glycan structures that contributes to biological diversity, but also to the speciation. And in fact, each cell in each organism, and I'm talking about we as mammalian or the microorganisms, the parasites, are covered with a unique array of glycans, a glycosignature that acts like an identity card that characterizes a bacteria or characterizes a virus or characterizes we as mammalian. But in fact, despite the glycosylation gene is quite well conserved across evolution, there are significant intra and inter-species variations that actually put glycans as an essential and a fundamental piece of information and as key molecular determinants able to distinguish self from non-self. Right? And actually, the first thing that an immune cell sees is actually glycans. As I mentioned previously, since all of our cells are are covered with glycans, the first thing that an immune cell can see to distinguish if this cell is from our body or is an intruder are glycans. So that's why glycans are an important piece of information in immune tolerance to guarantee homeostasis. What happens is somehow within a process that is called glycan mimicry, and glycan mimicry is the fact that there are, because of the evolution, there are some similarity of some glycan structures, some glycoantigens are somehow similar or shared between the host and the parasites. This is the glycan mimicry. And the glycan mimicry actually can be used by the microorganisms by themselves that can engage the glycan mimicry, decorating themselves with glycans from the host that are very similar to the host, and distracting the immune system, evading the immune recognition in order to assure infection and to perpetuate the chronic infection. But on the other side, the glycan mimicry can also confuses the immune system, and for this reason, the immune cells cannot distinguish self from non-self because of this glycan similarity. But this glycan similarity can occur also as a kind of a switch that may contribute to the glycan mimicry, and in this way to distract the immune system, leading to the inability of the immune cells to promote tolerance, to distinguish the self from non-self, and these will consequently result in autoreactive responses, in inflammatory responses, and autoimmunity.
First of all, I really liked your metaphor of glycans on the surface of cells being like ID cards. I've heard people also use, say they are like QR codes or they are like makeup, but essentially we are saying the same thing. So one thing I want to ask you before we move on is, you said there are certain similarities between the glycans in mammalian cells and in those of simpler microorganisms. In which ways do they differ?
During evolution, the complexity of the glycosylation machinery evolved. So mammalians and humans can actually synthesize very complex glycan structures. We are equipped with such a complex glycosylation machinery. We can extend and elongate the sugar chains. We can terminal sialylate them. We can fucosylate them. I mean, there is plenty of diversity in terms of the structures of glycans that humans can synthesize. But on the contrary, microorganisms such as the virus and bacteria are equipped with a less complex glycosylation machinery. They are not able to synthesize such complex glycan structures. And essentially, the glycans that they can synthesize are less complex, more primitive, not that extended and elongated, and they are mainly high manosylated glycans. But as I mentioned, there are some structures that are shared, some core structures that are shared between microorganisms and human beings that, of course, contribute to this process of immune tolerance, or in some situations, the inability actually to assure tolerance, leading to the process of the breach of immune tolerance.
As you said, glycans play a crucial role in recognition and tolerance, but they have been for decades very underappreciated when looking at immune regulation. Why do you think this is?
Yeah, so from many years, I would say decades ago, glycans were considered as simple, sweet decorations of the cell surface and were actually underappreciated for many years. But also, this is because due of the technological inability to go deeper on the structural characterization and the structural complexity of the glycans, that contrary to proteins, it synthesizes in a non-template driven way. But nowadays we are in the right moment, technologically speaking as well, but also, of course, all of the know-how that was gathered, the compelling body of evidences that were gathered along the years. So we are actually in a very nice and right position to tackle this complexity. The technology evolved a lot in the last couple of years, so we are able now to decipher the specific structure of the glycan, where it is attached in the protein, which carrier proteins are exhibiting and these glycans, where at the spatial level the glycans are located in a specific tissue. So yes, the technology evolved a lot, and this, of course, contributed to kick off and to foster this field of glycobiology.
Certainly happy to hear that. Now, moving on to thinking more about immune tolerance in terms of cancer. How long ago were changes in glycosylation first associated with cancer?
Yes, so the aberrant glycosylation in cancer was first described in the 60s or 70s, I would say more than 50 years ago. It was described some carbohydrate containing membrane components, and this was essentially, I would say, the first flag mentioning that, there are something beyond proteins, genes, and lipids that we need to account for the complexity of a cell, of a tissue, of organism. And of course, the consequences of these in cancer. And in fact, one of the hallmarks of cancer are actually the changes in the glycosylation profile. So the glycosylation profile of a cancer cell is completely different from the glycosylation profile of a normal cell. And this has been demonstrated along years of intensive research that these tumor-associated glycans have instrumental roles in each pathophysiological steps of tumor development and progression, from the very beginning of tumor initiation, invasion, but also in the metastasis process.
Now, thinking back on the ID card, as you said, the glycans change in the cancerous cells compared to healthy cells. So we would imagine the ID card also changes. And yet cancer is still able to evade the immune system. Why is this?
Actually, yes, this is one of the concept of immune evasion that give rise to this revolutionized field of research and clinical applicability of the immunotherapy that was actually the basis for the Nobel Prizes on that, because we know that in cancer and in the vicinity of the tumor cells there is a clear immune suppressive and immune regulatory network that allows and fosters cancer development and progression. And of course, glycans have been described by others and us to be important in this interplay between cancer cells and the surrounding immune system. What we demonstrated was the fact that there are specific type of glycan structures that has been widely described by others and us to be tumor-associated glycans with overexpression in a variety of epithelial tumors, such as, for example, the complex branched end glycans. And what we demonstrated was that this layer of complex branched glycans that are exposed at the cancer cell surface serves as a glycan shield that protects the cancer cells and hide them from the immune surveillance. And what we found was that when we removed this glycan mask, we were able to expose other immunogenic epitopes, allowing a proper immune surveillance by the immune system. So actually, glycans are essential interplayers between the cancer cells, but also with the immune system. And this is essentially because I think this is an important information. This interplay that exists between tumor-expressing glycans, tumor cells that express aberrant glycans and the immune system, and not only in cancer but also in inflammatory and autoimmune processes. This interplay with the immune system is essentially because immune cells are equipped with such a variety of glycan sensing receptors, and I'm talking about the cytoplactins, the galactins, and signacs that equip immune cells with the capacity to sense the glycosylation alterations, mounting either pro-inflammatory or anti-inflammatory responses. So essentially, this creates like a hub, a network between cells that express different types of glycan structures and the immune cells that are able to sense, detect, recognize, and instruct, depending on the glycans that they are sensing and recognizing, a pro-inflammatory response or an anti-inflammatory response. And if for example, if we're talking about the example of cancer, if an anti-inflammatory or regulatory response is mounted, this promotes cancer development. So if through glycans engineered or glycosylation reprogramming, we're able to promote the exposition of other immunogenic glycans recognized by the immune cells to mount a pro-inflammatory response, we will potentiate an effective and superior anti-tumor immune response.
Glycans in cancer and glycans in healthy cells do differ, and that does impact whether the pro-inflammatory and anti-inflammatory response is going to be stimulated. But do we understand the reason why there is a shift from a normal glycosylation pathway in these cancer cells?
What happens is that glycans are important, the protein glycosylation is important in homeostatic pathways. So they are important pieces, molecular pieces, to assure an homeostatic environment. And changes in glycosylation occur. And these changes in glycosylation occur in response to environmental or genetic stimuli that essentially leads to a glycan switch. In cancer, what happens, as I mentioned, there is changes in glycosylation is a hallmark of cancer, and these changes can be due to many factors. There are specific signaling pathways that are occurring in the cell associated with the cancer development process, and these signaling pathways are described to instruct the transcription of specific glycogens and to alter the glycosyl transferases profile in a specific cell, and this will consequently result in changes in the glycosylation profile. But there are other factors. For example, the immune environment and specific cytokines may also contribute to change the glycotranscriptomic profile in a specific cell, leading to consequently to changes in the glycosylation profile. Another example, for example, the pH, I would say the physiopathological conditions of the cell, for example, the pH of the cell can also contribute to alter the organization and the molecular assembly of the glycosyltransferases and glycosidases enzymes in the Golgi, and this will consequently have as an impact alterations of the glycosylation profile. However, it's a long-standing debate in glycobiology field and in cancer glycobiology whether the changes in glycosylation are a cause or a consequence of the cancer process. We have evidences supporting that changes in glycosylation can consequently result in cancer development, but we also have evidences that changes in glycosylation can be a consequence of a transformed phenotype. So we have both evidences supporting both events.
Now thinking of early detection and glycans being used as potential biomarkers of cancer, looking at your work with colorectal cancer, how early on were you able to spot glycans associated with cancer?
What we found was that these changes in the glycosylation profile of cancer cells, we have evidences that these might occur quite early in the process of carcinogenesis. For example, what we demonstrated and we published that was for the branched glycans catalyzed by the GNT5 glycosyltransferase, which is encoded by the MGAT5 glycogen. We found a gradual overexpression of these branched glycan structures along the colon carcinogenesis process. But it was very clear, the observation that the highest levels of abnormal expression of these branched glycans was observed as early as in premalignancy at the level of dysplasia, which is a premalignant condition. And this, of course, raises and opens a handful of many other questions about the relevance of these glycans and changes in glycosylation in early detection of cancer and in the prevention of cancer development. And that is something that we are exploiting in the group. Yeah.
Do you think cancer-associated glycans can be used as potential immunotherapeutic targets?
Yes, I mean, I think the concept of glycomedicine is gaining more and more and more power and impact in oncology, but also in other fields such as in immunity. Because, I mean, not only because, as I mentioned, there is this unique array of glycans that characterizes both an homeostatic but also a pathological condition, and this glycan switch that may occur associated with altered phenotypes associated with disease. So these can serve as glycobiomarkers. This signature that I mentioned previously, and we have evidences that not only, for example, in inflammatory bowel disease, but also in systemic lupus erythematosus, that the profile of glycans detected, for example, at diagnosis of a specific disease can have prognostic value, can actually serve as a biomarker for prognosis, for predicting, for example, response to therapy. The same holds true when we talk about serum levels of, for example, glycoproteins, and I'm talking about IgG glycosylation. There are more and more evidences showing that the pattern of antibodies glycosylation detected in serum can predict the development of inflammatory and autoimmune diseases. We have the example of rheumatoid arthritis. There was a seminal paper showing that there is a change in the glycosylation profile of IgGs that precede onset of rheumatoid arthritis. So not only as a source of biomarkers, but I truly believe that they can act as a target for therapeutic intervention. So the power of glycosylation reprogramming, envisioning to achieve homeostasis, I think it's immense. And we are, on the top of the iceberg, and of course we have plenty of opportunities to study the power of glycans as a source of therapeutic strategies, actually as immune therapeutic strategies, because the power of glycans to fine-tune the inflammatory response is very strong.
That's amazing to hear, because as someone who has autoimmune conditions, I know how limited our current therapeutic options are. But thank you for segueing us to autoimmunity. You have talked previously about how pathogens can take advantage of glycomimicry to confuse our immune system, and very often as a consequence people might end up with having autoreactive reactions. So do glycans on the tissues in our body, which are the target for autoimmunity, do those glycans resemble the ones on pathogens? Is there a similarity between those in people with autoimmune diseases?
We have evidence, for example, in lupus that it occurs specifically on lupus patients with kidney affection and having an inflammation in the kidney that we call glomerulonephritis or lupus nephritis. We show it that in lupus nephritis patients there is a clear glycan switch, and the glycan switch goes towards an array of glycans expressed at the surface of kidney cells that is very close to what happens in microorganisms. So we found a microbial associated glycosignature that, of course, engage in this process of glycan mimicry, and we have now evidences that these glycans switch towards this glycan mimicry. So there is a kind of a glycome signature that confounds the immune system and distracts them, leading to this breach of immune tolerance. And we have evidences that in fact this glycan switch promotes directly the activation of the immune system towards autoreactive responses. And we have that evidences in lupus, but there are of course other diseases that might occur as well. Actually, on that regard, we were quite recently funded by the European Commission under the Rise in Europe program with a project that aims to tackle the relevance of the mucosa glycome in the context of inflammatory bowel disease development, and the way that this mucosa glycome is relevant as a master trigger for health to intestinal inflammation transition. This is the Glycan Trigger project that we are just starting and sponsored and funded by the European Commission, aiming precisely to tackle the role of these glycocalyx and mucosa glycome composition as an essential piece that might trigger the health to intestinal inflammation transition.
No, that sounds incredibly exciting. What are the goals of this project, if you could summarize quickly?
Yeah, the goal is exactly that. One, so there is a clear shift between health and intestinal inflammation. This is associated with chronic inflammatory processes. Inflammatory bowel disease is one of them. So IBD, that comprises both Crohn's disease and ulcerative colitis, are two major chronic inflammatory disorders with a very important incidence in young populations in the active period of life. So essentially, we will decipher the role of mucosa glycosylation, the glycocalyx, as an early event that might promote and trigger a pro-inflammatory response, the way that it impacts the microbiota composition, the way that it triggers important hubs at the level of the intestinal mucosa, important immunological players that will be activated, leading to this transition process from health to intestinal inflammation, the way that this could be translated in a biomarker for disease prevention, and eventually as a target to prevent disease initiation. Yeah, that's the main goals of the project.
That sounds amazing. I'm particularly curious about thinking in context of relation to the microbiota in IBD. That's going to be very interesting to read about. But thinking of IBD and your previous work, what have you found already in terms of what happens to glycans in patients with IBD, and how early on can we spot these differences?
What we found on IBD was actually in the model of ulcerative colitis patients, we showed a clear glycosylation alteration, but specifically at the level of infiltrating T lymphocytes. So, essentially all of the chronic inflammatory diseases and autoimmune diseases are characterized by this huge infiltration of inflammatory cells, namely T cells. And what we found was that at the level of intestinal mucosa, we found a deficiency on a specific glycan structure on CD3 positive T cells. And these abnormal T cell glycoforms that infiltrate the mucosa were responsible for an hyperactive immune response that is associated with the severity of the disease. So essentially we show it also that we could metabolically glyco-reprogram this glycosylation profile of mucosal T lymphocytes, restoring, of course, the homeostasis and achieving immune control. And this, of course, also set the ground for the possibility of testing these in a clinical trial. Again, going back to the question that you raised about the way that glycans can be viewed as a source of agents with therapeutic applications. So in IBD, that's what we identified, and we also have evidences that if we change the glycosylation profile at the level of mucosa, this will consequently have an impact on the microbiota composition, and this is something that we will explore in a more deepened way in this Glycan Trigger project, which is actually a big consortium. We at the University of Porto will coordinate the consortium, but we have many other partners across the globe, I would say.
You have talked already about the potential of glycans as biomarkers as well as therapeutic agents, but just thinking of perhaps patient stratification, how well do you think glycans could be potentially used as predictors of therapeutic response in patients with IBD and as well as other autoimmune diseases?
We also have some evidences on that. So we show it that the levels of glycosylation, not only the levels, but also the abundance and the composition of the mucosa glycome detected in a biopsy that is collected upon colonoscopy has prognostic value. So we show it that for branched glycans, that was one of the glycan structure that we assessed and explored the potential prognostic value. We found that the levels of expression of branched glycans at the level of colonic mucosa in patients with an inaugural diagnosis of ulcerative colitis can predict the failure to standard therapy and therefore the eligibility to step up to more aggressive and powerful therapy, such as the case of biologics in case of IBD patients. So these, of course, highlight the power of these glycobiomarkers as important tools to be included actually in the clinical and therapeutic algorithm of IBD decisions. So not only clinically but also therapeutically. So one thing that we discuss it in based on the evidences that we gather is that if we so the way that the mucosa, colonic mucosa, is composed with glycans can inform the clinicians on whether to start early an aggressive immunotherapy would be better than to follow the step-by-step therapeutic intervention that starts in the classical anti-inflammatory agents and in a step-up approach. Increases the immunomodulatory power of the immunomodulation.
No, especially thinking of in terms of quality of life and just reducing the amount of pain these patients would have to undergo before the right therapy is found. That's amazing to think about. But now trying to circle back to cancer, is there an increased risk for individuals with an autoimmune disease to develop cancer based on your work?
And the other way around is also true.
Yeah.
So there is this fine, very tiny little line that separates, which is the tolerance, and separates what happens in autoimmunity, what happens in cancer, if we look to cancer as an immune suppressive environment. So what happens in cancer, and actually immunotherapy have revolutionized cancer therapy in the last couple of years. So essentially what we have in the cancer microenvironment is are networks with immune suppressive and immune regulatory response that actually allows cancer development and cancer progression, because there are strategies that are used by cancer cells to evade immune recognition and to contribute to the creation of these immune suppressive networks. So one of the strategies that are used that actually, transform the therapy in cancer is actually the immunotherapy, which is actually to remove the brakes of these T cells, of these immune cells, specifically the T cells, by inhibiting the immune checkpoint inhibitors. For example, PD-1, CTLA-4 are strong immune checkpoint inhibitors that suppresses the T-cell mediated immune response. And immunotherapy, the aim is exactly that, to remove that brakes, to potentiate an effective T-cell mediated immune response. But of course, these might fall in this tiny little line, and at the end we'll have an exacerbation, uncontrolled T-cell mediated immune response that may result in autoreactive responses. And actually, there is a not neglected proportion of patients under immunotherapy with immune checkpoint inhibitors cancer patients actually under immune checkpoint inhibitors that may develop and experience rheumatic and autoimmune diseases such as lupus, such as Crohn's disease. But the other way around is also true. So what we want in these autoimmune responses, and for example IBD, but also lupus, is it's the opposite. So we have an uncontrolled immune response, an exacerbation of the T-cell mediated immune response, and we need to suppress. And sometimes we are suppressing so much at such higher level that these may create the space and the condition, actually the spatial temporal condition for cells to transform, because the environment will become quite suppressive, regulatory, and cells take advantage and just transform, leading to cancer episodes. The same holds true. So the patients with autoimmune disease under these immune suppressive therapies may develop cancer. Yeah.
You've been mentioning the glycan switch quite a lot during this conversation, and I would like to just point to a recent ERC Synergy grant, which you and 3 other PIs have been awarded, focusing on the glycan switch in B cells. Could you tell us a bit more about that?
So the GlycanSwitch is a recent ERC Synergy funded project that besides me as a PI, we also have 3 PIs. So Gordon Lauk is another PI, but also Manfred Eurer and Tom Huizinga from the Leiden University Medical Center are the other 2 PIs. So it's all about glycans, of course, and the relevance in this process of tolerance. So the glycan switch is also as glycan trigger funded by the European Commission, but under, of course, in this case, the European Research Council. And with glycan switch, we aim to dissect whether the switch that we have evidences that occur at the level of B cells, and this occurs at the level of B cells preceding the development of autoimmune diseases such as rheumatoid arthritis. So we want to understand when, why, and how this switch on the glycosylation profile of B cells occur, the mechanism behind that, and the way that we could prevent the switch. And at the end, by preventing the glycan switch, we could prevent the development of rheumatoid arthritis and intercept disease development.
First of all, I want to congratulate you for both of these projects. It's very, very impressive. I want to mention for ERC, if I'm not mistaken, you are the 3rd Portuguese scientist ever to be awarded this grant. So it's an incredible accomplishment in your career.
Thank you so much.
If we could quickly just touch back on the glycan switches, you have a very interesting study looking at glycan switches in T cells in relation to COVID-19, and I think this is still a pretty current topic, sadly, 3 years later. But yeah, a little bit about that study.
That was our contribution as glycoscientists, of course, to understand the disease. So, I mean, the reason for our contribution is because we as scientists, we couldn't just stand by in the middle of a pandemic. And there was a national, in Portugal, a national, as in other countries, a national initiative by our research funding agency, to promote research in the field, and for all of the scientists that would be interested and with know-how could contribute, of course, to understand the disease and the virus and the pandemic. And actually, we took advantage of our know-how on glycoimmunology and specifically on T cells glycosylation And we decided to essentially screen a cohort of COVID-19 infected individuals, so SARS-CoV-2 infected individuals, and to see what happens to the circulating T cells in terms of the glycosylation profile. So, and making a long story short, what we observed was that infected individuals that develop a worse disease prognosis, so developing a severe disease, in comparison with those patients with a mild disease, have a different T cell glycosylation profile. So essentially, those patients that were able to switch quite efficiently the glycosylation profile of T cells towards a completely different glycosylation signature were more able to deal with the virus. And I mean, this was associated with a good prognosis, with a good disease course, in comparison with the others that upon infection with SARS-CoV-2, the T cells maintain quite, inefficient in terms of the glycan switch, enabling the T cells to efficiently deal with the virus. So essentially, we found that upon infection with the virus, there is a glycan switch, and depending on the efficacy of the switch, this is associated with the prognosis of the disease. Yeah, and that was our contribution to the field.
Thank you, Salome. I think we've definitely covered a lot of topics today. Is there a final message you would like to share with our listeners?
Thank you so much for the kind invitation and for these minutes of very nice conversation about glycans, which is something that I appreciate very much. Thank you.
I hope this conversation helped you better appreciate the nuances of immune regulation and the bright future of glycan prognostic and diagnostic biomarkers. If you would like to access more information about this conversation, as well as Salome's previous work, follow the link in the description to the show notes for this episode. Equally, if you would like to find out more about GlycanAge, head on to glycanage.com, where you can access a whole list of our scientific publications, blog posts, testimonials, and of course this is where you can order your GlycanAge kit. Watch out for our next episode, where I will be joined by Olga Zaistova, a geneticist focusing on genetic regulation of immunoglobulin G glycosylation. We will discuss the genetic and epigenetic control of IgG glycosylation and, by extension, chronic inflammation, especially in the context of behavior and lifestyle. Please don't forget to leave ratings and reviews for this episode and engage with us on social media. All of the links will be in the description. Thank you for listening and have a great day.